Now that we know how a cell divides, it's important also to understand when. Not all cells go through division once they are created. Some cells just hang out as the particular type of cell they're meant to be, and they function in the body, and that's it, and they never divide again. Whereas other types of cells will continuously proliferate to help sustain homeostasis in, for example, a multicellular organism. So really the frequency of cell division is going to vary with cell type, and it all comes down to what kinds of molecules do those cells express. Do they make the molecules that allow them to go through cell division, or do they not? Now, there are also cells that have lost control of their cell cycle and they don't know when to stop dividing. These are cancer cells. And so we're going to spend a little bit of time at the end of this chapter talking about cancer, how cancer cells we think maybe lose control of their cell cycle, and then how that contributes to the pathology of the disease. So let's talk about the cell cycle control system. It's the cell cycle is really driven by a variety of different molecules that interact with each other within the cytoplasm of the cell. There's no one, structure within the cell that controls this. It's just simply, an interaction between different molecules that helps to turn on certain processes or keep certain processes turned off. And so the sequence of events during the cell cycle is controlled by the cell cycle control system, right? I like to think of it kind of like a dial on, an older model washing machine. You've got the little dials where, you set it to the particular cycle that you want. It has to click through, the wash cycle, and then the spin cycle, and the rinse cycle, and then another spin cycle, to get through the entire wash process. It's kind of the same thing for the cell cycle control system. The cell has to complete G1 before it goes into S, and has to complete S before it goes into G2, and has to complete that before it goes into the mitotic phase. So we're going to use this sort of, washing machine dial analogy as we discuss the ways in which the cells will go through the cell cycle and go through these certain checkpoints to make sure that the cell is in the right sort of state before it moves on to the next stage within its life cycle. And the cell cycle is controlled both by internal and external controls. So cells can respond to extracellular signals, or they can respond to things that are going on in the cell itself that will help it move through the cell cycle. And this cell cycle control system has these specific checkpoints where the cycle stops, it stalls, and then it waits and it looks for, it senses for certain signals that will allow it to move past the checkpoint, the sort of go-ahead signal. If it doesn't get that go-ahead signal, it stays at that checkpoint stuck, essentially, and it doesn't progress further. So here's a cartoon diagram showing you kind of what we're imagining as this, analogy for the cell cycle control system. So you've got the full cell cycle, you've got your interphase G1, S, G2, and then you've got the mitotic phase down here. And so you can see a little control system. It's like a little dial, and it'll click through the different parts of the cell cycle, and then it gets stuck at these red barriers. These are going to be the checkpoints. This is where the cell stops and checks to make sure everything's in order before it moves on. And so we're going to talk about these three important checkpoints. There's one here in G1, one in G2, and one right in the middle of the mitotic phase. This is going to be between metaphase and anaphase. The first one we're going to talk about is the G2 checkpoint. This is the most complex out of all of them. This is an example of where an internal signal helps to regulate the cell cycle. The G2 checkpoint is regulated by a pair of proteins. One member of this pair is called a cyclin. A cyclin is a protein whose concentration varies, goes up and down, along with the different phases of the cell cycle. The other half to this partnership is what is called a cyclin-dependent kinase. You've probably never heard this term before, but hopefully you can glean some information from the name, right? This last word, a kinase, it ends in ASE, should give you the hint that, hey, this is going to be an enzyme. Specifically, kinases are enzymes that can phosphorylate other molecules. They can add phosphate groups to other molecules, primarily probably enzymes. Remember, we talked about how phosphate groups can be the on-off switches. You put a phosphate group on a protein, you turn it on. You take the phosphate group off of a protein, you turn it off. And so kinases, by phosphorylating other enzymes, can initiate this process of going into mitosis. All right, and so if this is a kinase, it can phosphorylate other substances. More specifically, it's also a cyclin-dependent kinase, so it will only be active when its cyclin is present. And if the cyclin's concentration goes up and down with the cell cycle, the activity of the CDK, or the cyclin-dependent kinase, will also rise and fall with changes in the concentration of the cyclin partner and changes in the cell cycle. Now, there are lots of different cyclins and cyclin-dependent kinases, or CDKs, but there's one important pair that's called MPF, or the maturation promoting factor, which helps to regulate this G2 checkpoint. If the maturation promoting factor is present and active, then the cell can effectively leave interphase, G2, and enter active mitosis in the mitotic phase. Let's take a look at an example. All right, so here we've simply got your cell cycle in the middle, right? G1, S, and G2 of interphase, and then we've got the mitotic phase in yellow. Here's our G2 checkpoint. So you'll notice that throughout the cell cycle, this orange CDK is always present, right? So the kinase is always available. On the other hand, the cyclin, a little purple half circle here, cyclin concentration isn't always the same. Cyclin concentration starts to accumulate towards the end of interphase as the cell anticipates going into the mitotic phase. As cyclin concentration goes up, more frequently it will bind to its CDK, forming the complete maturation promoting factor. The kinase is now active. It will go on to phosphorylate other enzymes, initiating the mitotic phase. Once the cell is in mitosis, it no longer needs activation of the process, right? It's already in there. And so the cell will degrade the cyclin. This leaves the CDK by itself and inactive, and it just sort of, sits around, hangs out, and waits until the cell goes through the cell cycle again, if it chooses to. And it waits for its cyclin concentration to accumulate again so that it can become active once more.